This week I'd like to talk about genetic engineering, mostly the case of human genetic engineering. So in this video I'll lay out some of the background and the kind of landscape of the debate, and in the other video I'll talk more specifically about the readings for this week. So in terms of how ethics and values should impact science, which is kind of the topic that we're going to shift to for the remainder of this course, I think it's hard to think of something more urgent than genetic engineering. Literally, the future of humanity is at stake here. What kind of species we're going to be going forward are the stakes of this debate. It's a question of who we are and who we want to be. Like what's going to happen to Homo sapiens in the next 100 or 1,000 years. That's what we're discussing here. So this is an absolutely central question in the sort of science and values world. Before we get into talking about gene alteration by humans, I'd like to lay out some of the kind of background biology and in some sense explain why, at least I personally, I'm not worried about the very idea of changing genomes. I'm worried about a lot of aspects of genetic engineering, but that's not one of them, and I'd like to explain why. So it's easy to think of DNA as a very static molecule, right? So the DNA that you have is generally speaking the DNA you were born with, and if you reproduce, you'll pass on some version of it, or at least half your genes, to the next generation. But it's actually quite a dynamic molecule. Quite a lot goes on with it throughout your lifetime. So, for example, lots and lots of stressors can break the DNA strand, and those breaks are almost always repaired within the cell. By some estimates, your body repairs as many as 1 million breaks per cell per day. So the DNA is not just sort of sitting there perfectly static. It's being broken up quite a lot, and your cells are constantly having to repair it. Adding to that, while we usually think of DNA as strictly coming from our ancestors, in many ways that's not always true. So for example, there's what's called horizontal gene transfer. So vertical gene transfer is where you get it from your parents or whoever contributed your DNA. Horizontal gene transfer is what happens when genes can be transferred between unrelated living things and sometimes between species. In simple single-celled organisms, horizontal gene transfer can account for up to 20% of their DNA, 10 to 20% of their DNA has been transferred to them, not from their ancestors, but horizontally between organisms. So what does that even look like? In single-celled, simple organisms, this is often in the form of what are called plasmids. So a plasmid is a little ring of DNA. If you can see the picture, it's this little blue ring. So it's a little circular bit of DNA that can be released from the cell and absorbed into another cell, sometimes actively transported into another cell. It's not too far off to think of these plasmids as like little apps that can be traded between cells. So these little ring DNAs are separate from the kind of main chromosome of the bacterium, but they're traded between bacteria, and they can confer things like immunity to certain viruses or diseases or cellular abilities, things like that. So single-celled organisms, at least, are trading these little plasmids all the time. Horizontal gene transfer was, for a long time, thought of as just a phenomenon of very simple life. But in the last 20 years or so, it's become clear that that's not true. In multicellular life, in plant life, we also see horizontal gene transfer. So it's actually fairly common in plants. We're not completely sure how this works. There's a number of mechanisms by which plants might trade genes between each other. Some of the mechanisms proposed have been things like parasitic plants. So one plant has a parasite living in it, and that kind of intimate contact allows them to trade DNA between both the parasite and the plant. You might have transfer via bacteria. So a bacterium takes a bit of DNA from one plant and then goes and lives on another plant and gives that DNA to the other plant, or what they call just naked DNA, just sort of DNA strands in the soil being taken up by the roots and incorporated into the plant's biology. So there's a debate about how prevalent this is, how important it is to the sort of evolution of plants, But the reality is that horizontal gene transfer does seem to be a reality for even some forms of complex life. It seems to be relatively rare, at least compared to plants, in animal life, but it's not completely absent either. So horizontal gene transfer seems to be a way to get a different set of genes than the ones that you were born with. So since the 1970s, scientists have been able to transfer genes from one living thing into another. Call it recombinant DNA technology. It's recombinant because you're recombining the DNA from one living thing into a different living thing. The flagship example of this is probably taking the genes from certain jellyfish, which cause them to glow in the dark. They phosphoresce. And if you take those genes and put them, say, into a plant, you can make a glow-in-the-dark plant or a glow-in-the-dark mouse. So this is profoundly worrying to some people. Recombinant DNA technology is, I think, not without any reason, profoundly disturbing and worrying to people because they think of DNA as an exceptionally stable molecule, right? It's stable over the level of generations, millennia, eons sometimes. But what I just told you about horizontal gene transfer and the kind of dynamic character of DNA is why at least I personally don't worry about at least just the idea of changing the genes in a plant. The genes that are present in living things seem to be much more dynamic than we thought they were, say, 30 years ago. What does worry me is perhaps the way that recombinant DNA technology gets used. So one of the most famous examples is what are called Roundup Ready crops. So Roundup Ready crops are a kind of crop that's been genetically modified by putting genes in that make them resistant to a certain type of pesticide called Roundup. So Monsanto sells this pesticide called Roundup, and they also sell seeds that have been genetically modified to be able to handle more of that specific pesticide. So you can really lay it on your crops, really kill any of the pests that would be there, and the crops don't die because they've got these genes that make them resistant to the Roundup. So you might worry about that. So that's not for me, when I see a little, non-GMO sticker on a box of cereal or something like that, my mind doesn't go to worrying about novel genes in the corn that went into making the cereal, because I know that corn has been undergoing horizontal gene transfer for a long time. Its genes have not been static over the millennia. They take in new genes on a natural, fairly regular basis. What does worry me is that somebody's made that corn specifically able to handle really large amounts of pesticides. So it's the use of the recombinant DNA technology that worries me personally, not the technology itself, not the idea of changing genes. That's my opinion. You might have different thoughts or feelings about that. This is the debate that's ongoing about sort of the simplest case of genetic engineering. Okay. So that's been ongoing. Recombinant DNA technology has been ongoing for more than 20 years. I believe Roundup Ready crops were around since the mid-90s or something like that. Let's talk a little bit about the kind of cutting edge of genetic engineering technology, and that's undoubtedly the CRISPR technology. So CRISPR refers to a kind of DNA sequence found in bacteria, which form part of their immune system. I won't get too deep into the weeds of the details here, but in 2012, a team of scientists discovered that they could use an enzyme called CRISPR-Cas9 to make cuts in DNA in very specific places. So they're using an enzyme that's present in bacteria that's used for the bacteria to sort of defend themselves, and using it to make very careful cuts, very precise cuts at a specific location in the genome. And then once they've made that cut, what I just told you about, the sort of cells repairing their own DNA, the cells will attempt to repair the cut. Now, if they are left to their own devices, they'll just try to repair it with the sequence of DNA that was there. But if you also introduce another sort of gene sequence into the cell, sometimes it will repair that cut with the gene sequence that you were hoping to get in there. So this is a way of introducing a new genetic sequence into a very specific part of the genome that you're looking to intervene on. Okay. For our purposes, the details don't matter too much. The metaphor that I've heard for this is that it's sort of like going from having to kind of, if you want to change the content of a book, a printed book, you got to reprint the book, right? This is a lot like going from that to being able to kind of cut and paste into a Word file, right? So we've got this much more dynamic access to the genome as of 2012 and going forward the last 8 years or so than we ever did before. Our ability to change the genome has increased massively over the last, say, 10 years. And I think it's interesting to note that a bunch of scientists, including Jennifer Doudna, who was one of the scientists who actually developed the CRISPR technology, have been a bit worried about this. So in a paper published in Science in 2015, a group of scientists, including Jennifer Doudna, called for extreme caution in the use of this technology. They say, quote, "At present, the potential safety and efficacy issues arising from the use of this technology must be thoroughly investigated and understood before any attempts at human engineering are sanctioned, if ever, for clinical testing." So it's not common for scientists to weigh in on these kind of policy issues about how their discoveries get used. Jennifer Doudna and her collaborators won a Nobel Prize for this technology in 2020. And they have come out publicly, and other groups of scientists have come out publicly, suggesting that a broad public conversation needs to happen before it's reasonable or responsible to use this technology, especially on humans, and especially on what I'll explain in a second is the germline in humans. So this is not a thing that happens a lot. That the very scientists who develop a technology warn humanity about its use. So I think that's worth noting and taking very seriously. Note that they're not saying that we should never ever use this, and they're not just making a claim about its safety. They want the sort of broader discussion amongst scientists who know this stuff very well is they would like for there to be a broad public discussion, not just expert, but non-experts as well, about how we think it's reasonable and responsible to use this technology before we start using it, because the effects might well be permanent and the implications might be deep and profound. So, for context of this discussion, I think it's worth noticing that humans have been changing ourselves for as long as anybody can remember. Most cultures, every culture that I'm aware of, in fact, I'd be curious if you can think of an exception to this rule. There probably is one, but every culture that I can think of has some tradition of modifying their bodies, from tattoos to circumcisions to sort of lip plates. The vast array of ways in which we modify our bodies. You can hardly dig up a corpse from a bog. When you dig up a 3,500-year-old body that's well preserved, you see dozens of tattoos all over their bodies. So there's this amazing array of ways in which humans modify their own bodies and have done for pretty much as long as we've been human, as far as I can tell. It's amazingly widespread. It seems to be something really deep in us, or at least something that's extremely common and extremely important to people, the sort of ability to change our own bodies. And I don't know of any other animal that does it, especially to the depth that humans do this. So it seems to be a characteristic that's somewhat unique to humans, or at least its degree of expression is unique in humans, and it seems to be extremely widespread amongst humans. We are the animal that changes ourselves. So I don't think it's reasonable to come out against changing ourselves in general, given that fact. However, there's something quite unique about genetic engineering, and especially a certain type of genetic engineering. So let's distinguish between somatic and germline genetic engineering. So somatic just means you're an adult and you change the genes in your adult body, but not the genes in, for example, your sperm or your eggs. So germline genetic engineering is genetic engineering that would change the next generation and the generation after that and after that. So germline genetic engineering has a kind of consequence that no tattoo or body mod or anything like that has ever had before. It's not like when you get a tattoo, your kids get that same tattoo automatically, right? So we're talking about alterations to all of the future generations that might come out of your body, right? If you're going to reproduce, every generation after you will carry that modification if it's a germline modification. Now there are somatic alterations that don't get passed on, so you might feel very differently about those two types of genetic engineering. You might worry quite a bit more. You might be much more cautious about altering the germline than just altering the adult sort of genetic structure. Okay. I think at the very top of our minds in any kind of discussion, especially about some germline genetic engineering, should be the quite horrifying history of eugenics. This isn't the first time people have talked about shaping the human genome. And the last time we talked about it, it was bad. It was really bad. So the extremely ugly history of eugenics need to be, in my opinion, front and center in our minds whenever we have a discussion about shaping the genome. So recall this fella, Francis Galton. He thought that some people that he and others deemed undesirable should be prevented by force if necessary from reproducing, and the people that he considered desirable should be encouraged to have more children to sort of alter the structure of the human gene pool. And this led to things like the Holocaust, for example. I mean, just off the top of my head, the Holocaust comes to mind. Forced sterilizations that continue up to this day in this very country, and were officially sanctioned as eugenics in Alberta up until the 1970s. This is an extremely worrying. So the combination of, hey, let's tinker with the gene pool, and even the hint of racism, if not full-blown racism, is a deadly, deadly combination. So this is something that we should, at minimum, kind of keep in our heads when we're thinking about genetically altering ourselves. And one of the sort of discussions that's ongoing right now about genetic engineering is sort of focuses on disability. So communities like, for example, I mean, a bunch of communities, but for example, the deaf community or the autism spectrum community are really worried about people who are, say, offering to cure them of their condition using genetic engineering. Communities like the deaf community and the autism spectrum community have been very clear, or at least there have been very strong voices in those communities saying, my state of being is not a disease looking for a cure. People in the autism spectrum community, for example, say, look, I'm different than you, but that doesn't mean that I'm less than you. That doesn't mean that what I am should be cured. Yeah, I don't have a disease. I'm just different. So their alternatives is, of course, that the world should be structured such that there's room for people who are different than the average. That's their alternative pitch is, don't try to wipe me out with genetic engineering. Make a little space in the world for people who are a little different, and the world will be richer for it. So when we're thinking about this stuff, again, and especially in the context of keeping in mind the kind of the looming threat of eugenics, when you're offering to cure people of something, you might wonder, and you might seriously ask yourself, is this something that is a disease that needs cured, or is this just human variation? So one of the threats of genetic engineering is that we treat natural human variation as though it's a disease, and sort of chop off any differences that don't fit into modern neoliberal capitalism in order to make the world sort of run better according to one picture of how it should run. So these are things that, I mean, you can imagine, for example, somebody offering, we'll identify the gay gene. This is not a gene that anybody's identified, but identify the gay gene and cure it. And I think most people in the homosexual community or the queer community would say, I don't have a disease. You can't cure something that's not a disease. So these are the kind of concerns about the way in which genetic engineering might be used. Compounding this are concerns about inequality. So if you've seen Gattaca, I mean, this is certainly the kind of major theme of that movie. if genetic engineering is expensive, then only people who can afford it will get it. And that might lead to, and if it genuinely helps people be more functional, or by some definition of functional, that puts increasing sort of like inequality into play. So if you can genetically engineer somebody to be more likely to become rich or to stay rich, then the inequality will be exacerbated by genetic engineering. So concerns about nascent eugenics, concerns about trying to cure things which aren't diseases, and concerns about driving inequality are all things that you might consider as worries with respect to how genetic engineering might be used. Okay, so with all that in mind, let me just mention that CRISPR technology is now being used on humans. So we are using CRISPR technology on humans as of now. In 2020, at the Oregon Health and Science University, a clinical trial was done to try to repair a gene which causes blindness. So it's an inherited gene, it's not just a random mutation, and it tends to cause blindness after childhood. So it's worth noting that this is it's a somatic gene therapy. It's not a germline gene therapy. So the changes that are being made aren't ones that would be passed down. But this is the thing that we're doing. I think it's worth noting. Okay, so to wrap up, I think nobody in the discussion thinks it's reasonable or ethical to deploy this technology until we're pretty confident that it's safe. But scientists are hopeful that it will be broadly applicable to a range of conditions. And I've just gone over some concerns that you might have. Supposing that it's safe, there are further concerns, which I've just been discussing, about its application. In the next video for this week, I'm going to go over the readings for this week, and I'm going to present some arguments from biology suggesting that we might not be as close to a safe and side effect-free version of genetic engineering as it might look. And specifically, we'll talk about whether the engineering metaphor is appropriate in this domain. Okay, thanks.